Hello, everyone, and welcome to our next session. This session is entitled The Genetic Architecture of Autism in an East African Cohort. It is my pleasure to introduce to you Maria Chahrour and Leah Seyoum-Tesfa. Maria is an associate professor at University of Texas Southwestern Medical Center, and Leah is the founder and director of Reaching Families Advocacy and Support Group. You can reference your attendee website for their full bios. On the next screen, we'd like to share with you and point your attention to the CEU accreditation and disclosure statements. Our speakers for this session do not have any disclosures to report. You can also see their learning objectives listed here. With that, I'd like to turn it over to Maria and Leah to present your slides. Take it away, ladies. Thank you for being here.
All right. Hello, everyone, and thank you for joining us today. And thank you to the organizers for the invitation to speak here. Today, I want to share with you an overview of the work that we're doing in the genetics of autism space, focusing specifically on one story on our autism gene identification efforts in collaboration with the East African community here in Dallas. So before I dive into the talk, I'll start with a very brief introduction and then define how we think about autism from the genetic and molecular perspectives. As the name suggests, autism is a spectrum of neurodevelopmental conditions characterized by deficits in interactions with society and the environment. It affects up to 2.8% of children, with more males being affected than females at about 3 times more. And the prevalence of ASD has been increasing at a rate of about 7% annually for the past 20 years, posing significant societal and economic burdens, more than the cost of cancer, heart disease, and stroke combined. In addition to the core symptoms of impaired communication skills and social behavior abnormalities and repetitive behaviors, there's a host of comorbidities, or these are additional conditions that also occur alongside autism. And these include behavioral, psychiatric, medical, and developmental comorbidities. The numbers here show the proportion of ASD patients who also have these comorbidities. And some common examples include intellectual disability, motor function, seizures, and anxiety. ASD is very complex in the way that it presents clinically, but what causes it? We know from many large twin studies that ASD is highly heritable, with a heritability of up to 90%. And what that means is that changes in your genes account for whether you have ASD or not. And human genetics research over the past 2 decades has identified hundreds of ASD genes, and a proportion of these are associated with well-defined syndromes such as Fragile X, Rett, TSC, and Angelman syndrome. And another proportion of cases are due to chromosomal abnormalities, or what we call copy number variants. And there are several hotspots for these CNVs in the genome that are indicated here, including the 16p11.2 locus and others. Another proportion of cases are due to spontaneous, which are also known as de novo mutations, and another proportion are due to inherited point mutations. And I've listed some of the common genes here as well. Finally, some cases are due to somatic mutations, and these are changes in the DNA that occur only in the brain. And some somatic mutations have been identified in known ASD genes. Now, for the majority of cases, about 65% or so, the underlying genetic causes have not yet been identified. And although I've told you that hundreds of genes have been identified, each of these genes on its own contributes to a very small proportion of individuals with ASD. So I've listed here some examples. For example, the mutations in the Fragile X gene, FMR1, account for up to 2% of cases, or 1.94%. And then the next gene, TSC, accounts for less than 1% of cases, and so on. You can see each of these contributions listed here. So because of all the data from the human genetic studies to date, we can start thinking about ASD as this collection of individually rare genetic subtypes. And in order to start understanding the pathophysiology of ASD, we have to understand the molecular mechanisms underlying each of these different genetic subtypes so that we can start grouping them, for example, based on specific pathways that are disrupted, and we can start thinking about therapies. And also importantly, so we can inform genetic diagnosis. Of course, when you think about ASD as a collection of individual genetic subtypes, that doesn't mean that you should think of therapies for one subtype at a time exclusively. You may need to do that for some genetic forms, but for others, you may be able to combine the subtypes based on deficit in a common brain circuit or pathway, or even an affected brain region. My lab at UT Southwestern is interested in identifying ASD genes, studying their function in the brain and the molecular mechanisms that they regulate, so we can use this information in diagnostics and the design of therapies. And I'll give you an overview of our ongoing genetic studies and how we identify ASD genes and investigate their function, focusing on one specific story for today on our collaboration with the East African community in Dallas. So I'll start with sharing an overview of the human genetic studies that we've been doing. And we identify disease genes through whole exome or genome sequencing in families with ASD. And we have protocols approved by the UT Southwestern IRB to recruit and enroll participants to perform these studies. We partner with our community and the families to include samples from ancestrally diverse participants and also participants across the ASD spectrum. And we also collaborate with our clinical colleagues to identify interested study participants and obtain detailed clinical information and medical history. Our goal is to identify ASD genes and ultimately inform diagnostics and therapies, and in doing so, to learn about brain development, social behavior, and language. Our study so far has enrolled 3 cohorts: a local DFW cohort, cohorts with shared ancestry, and an African cohort. And to date, we've enrolled over 3,000 participants, and we're still actively enrolling. As I mentioned today, my talk will be focused on our study with the 3rd cohort, an East African cohort of families from an immigrant community here in Dallas, that we enrolled through partnership with members of the East African community and Reaching Families Advocacy and Support Group, which was founded and is directed by my collaborator, Leah Seyoum-Tesfa, and you'll hear from her shortly. So our motivation for this study is twofold. One is to understand the increased prevalence of autism in the East African population, which is about 5%. And we did this by leveraging ancestral information and genetic diversity of their African genome. And then the other motivation for our study is to bridge the diversity gap in human genetics. As some of you may know, the majority of studies to date have been performed almost exclusively in populations of European ancestry. And for the first time, we investigated the genetics of ASD in an African cohort. Importantly, after we identify ASD genes and variants through these concerted efforts, we are really interested in understanding their normal function in the brain so that we can pinpoint the specific pathways that are affected and the mechanisms that they regulate. So before I go into the details of our project, I want to just give you a little bit of background. So according to the WHO, the prevalence of ASD globally is about 1%. And of course, this number doesn't take into account the prevalence in many low-income and middle-income countries, because the prevalence in many of these countries is not known because these studies are not being conducted there. The reported prevalence is also very geographically variable within the United States itself, and that's because it's based on access to assessment, diagnosis, and services. And so far, the majority of the global prevalence studies have been done focusing on the United States, North America, Europe, East Asia, and Australia. And then for the entire continent of Africa, there are only 4 prevalence studies that have been reported so far. 1 in Libya, and 1 in Nigeria, 1 in South Africa, and 1 in Uganda. So there's definitely a lack and a major need for conducting these prevalence studies on the continent of Africa. So before I get into the details of the study, I'm going to hand it over now to my collaborator, Leah, so she can tell you about her part and listen to it from her lens. So Leah.
Hello, thank you for the opportunity to present, and thank you, Dr. Maria Chahrour, for the opportunity to collaborate with you on this project. These young, beautiful men that you see, they're 25 years old now, are my children with autism. I have 4 children, 2 of them, they are twins, were diagnosed with autism when they were 3 years old. I come into the world of autism, and I am by training a nurse, and I struggled to find resources early on for my children and to advocate for them, to learn the whole world of autism and how to support my children. So as they got older, I started looking into my community. I'm originally from Ethiopia, and my parents are actually Eritrean. So I was very much in that community when my kids were diagnosed, and I looked around. There was a lot of children like my children that were being diagnosed with autism, and a good number of them tended to be on the severe end of the spectrum. And I started to support families, advocating and helping them through the system, resources, and teaching them how to work with their children. So in 2011, I ended up starting a nonprofit organization to support families in East African families. Currently, we have 25 support groups all over the United States, Europe, and Canada. The support group, it's an advocacy organization. We provide education, empowerment, advocacy, mentorship, grant and scholarship for some families who are low income. We do fundraising through the community, and we also have received grant from the state of Texas in the work that we do. We work with the community. We go to the churches to do outreach. There's a lot of stigma around having a child with special needs, encouraging parents to access early intervention, teaching them about the science of autism. The organization does not work only with children with autism, but any child with developmental or physical disability. So when working with this and with the Ethiopian Eritrean families, I started to realize that it seems like a lot of our kids, there's more kids that are affected with autism in our community than other communities around us. As I was exploring this and the number of families that kept calling us for help, and as I was going to the churches and seeing a lot of the kids in the Sunday school programs seemed to be on the spectrum. This study came out in 2013. This is from the Somali community. Somali is a country right below Ethiopia. We share a lot of genetic backgrounds with them. And the Somali community in Minnesota is very strong. There's many Somalis that live in Minnesota. And they wondered the same thing that I was wondering. They had a lot of children in the school system, and the parents were saying a lot of their kids had autism. So this project was done by Minnesota Public Health, Minnesota, Indianapolis schools, and CDC was involved in this report. And they reported that 1 out of 20 kids, males in Minnesota, and actually the study was in Minneapolis, 1 in 20 had autism, were diagnosed with autism. And as you look at this chart, it shows that in the white population, 1 in 23, which seemed very close. But as you see in the Hispanic population, a lot less. And overall, 1 in 30 male had autism. When we looked at this study, it came out in 2013. The Somali children that were identified, 100% of them had IDD. As far as the white children had 1 in 23, I believe. If you can go back to the slide, I think it was 1 in 20 had intellectual disability. That this study for us working in the community shows that the higher end kids that are not on the severe end are usually not identified in these communities because of the stigma, because of the parents not knowing that when they see autism, usually it is the nonverbal, the more affected children are usually identified. So this tells you that even with the only the portion of kids identified, the rate in the Somali community was very high. At that time, we can go to the next slide. At that time, I had the opportunity to participate in the Lone Star Lend Fellowship Program in Houston. It's a program for people, parents, and professional medical professionals of a one-year fellowship program that we learn about the different aspects, resources, and how to work with families who have children with neurodevelopmental disabilities. As part of my fellowship, I did research looking at the data in our community. So this was, it's not a published study, but I did report on this. It's prevalence of autism in children of East African descent in Texas. The data was provided to us from Texas Educational Agency, and we looked at children all over Texas and who were in the public school system, who were identified as having autism. We were able to identify whether they were in our community by the language survey that parents need to fill out when they enroll their kids. Now, as you know, public schools, everything is not, everybody does not fill up the forms, but for those that were, and some parents also have some hesitancy to fill up the form, letting people know the children are exposed to another language. We did see also a higher rate of autism. In Texas, at that time, it was 1 in 167 children. In our community, we saw it depending on what language we looked at. Ethiopic was 1 in 22, and Amharic was 1 in 40, that's the national language, and 1 in 51 in Tigrinya, which is a language spoken up in the northern. There are over 72 languages spoken in Ethiopia, but these are the most popular ones at that time that were actually, I was able to scan and identify as the school districts, giving parents a choice to fill out. So we did see again then that there was a higher prevalence in our community. And that is, this picture here is the families that I support at that time. It's a portion of them that I support at that time. And this is also the families that participated in the study, some of them. So, and then I looked at the research. Part of what I did in reporting the research is I looked at what other research was out there in our community. I was surprised to find out in these studies were already done. They were looking at prevalence or if there are higher incidences of autism in children of African-American children. Most of these were immigrant children. One study was done in Australia, and they did see 3.5 times more African-American children, East African children were identified in that study. The study in Los Angeles was done, and they did identify that immigrant families that had children with autism tend to have more significant higher incidence and more significant disability. There were also a study done in Ireland, in Sweden, in Canada. There was a study in Israel. Also, there was immigration of Ethiopian families to Israel in 2004, and they did see a higher incidence of children from Ethiopia. And there was the oldest study was in Sweden, and they did see, that was in 1987. They did see that immigrant families, what they called exotic countries, tend to have higher incidence of autism. So the literature did show that there is higher incidence in our community.
Okay.
Yeah, so after that, I think there should have been a slide here. And then after I looked at this, and we were able to connect with Dr. Chahrour, who was already doing a study on autism. So we communicated, and we came up with a study to look at our population, and a lot of the recruitment was done by my organization, and most of the participants were in the DFW area, but a number of them also were from other parts of the world, and I think there's one family that was from Europe. So that's the study was done, and I'll turn it back to Dr. Chahrour.
Thank you, Leah. Yeah, so as Leah told you, there was evidence from her own study that she did in Houston through the public schools in Texas, and also all these other studies in the literature that had reported a higher incidence of autism in East African families, and specifically in Ethiopian populations. And so just to summarize it here, that, with all this information and the data about this increased prevalence, that suggested to us that there was a higher incidence in East African populations, that there's potentially a shared genetic susceptibility factor that's predisposing to autism. And so we wanted to ask, what is the genetic contribution to the increased autism prevalence in these children? And this is how we started. But before I go into those details, there's still one additional piece that I want to mention. And I had told you a little bit about, one of the drivers being that we wanted to leverage this genetic diversity in the African genome. And populations in Africa are the most genetically diverse in the world and carry up to 3 times as many rare variants as populations of European or East Asian ancestry. So the plot that you can see here on the left shows the genome-wide heterozygosity, and this is basically a measure of genetic diversity in a genome, that it's highest in East Africa and that it decreases worldwide as you get further away from Addis Ababa and Ethiopia, which is in East Africa. And then the plot on the right-hand side shows that the percentage of rare variants in different populations, with the African populations having the highest percentage of rare variants compared to European or East Asian populations. And to date, unfortunately, there's no comprehensive catalog of African genetic diversity. Although there are multiple ongoing efforts, for example, the Human Heredity and Health in Africa, HD Africa, and other efforts to generate this catalog. So in the first publication from the African Genome Variation Project, which is this international collaboration that's aimed at characterizing African genetic diversity, the AGV found that the Ethiopian population has the greatest proportion of all novel and private genomic variation found in Africa, about 24% of genomic variants. So with this information of increased ASD prevalence in East African populations and the genetic diversity of the African populations, our strategy for this project was to leverage the ancestral genomic information so we can identify ASD genes by combining whole genome sequencing and admixture analysis in a familial cohort from a population with an unusually high rate of ASD. And as I mentioned, this is one of the first studies to investigate the genetic underpinnings of ASD in an African population. And it started from an organic collaboration between myself and Leah, and we're partnering with members of the community here in Dallas through her organization, Reaching Families Advocacy and Support Group. And we're continuing our study to expand it and enrolling additional families. So how we did this, we recruited East African families, and the parents who enrolled in our study were all born in East Africa, predominantly Ethiopia and Eritrea. We had one family from Kenya, and the children were all born to East African parents. So there's no outside admixture for a human genetics analysis purpose. And all of the affected children were diagnosed with ASD. The vast majority had speech abnormalities, and there were also other comorbidities in the cohort that are typically associated with ASD that you can see here. And as Leah mentioned, this recruitment was done largely through her organization here locally, but also through her connections with communities pretty much internationally. So we did whole genome sequencing, and just to give you a little bit of perspective, the human genome has 3 billion base pairs or letters, And we share 99.9% of our genomes, all of us. So this 0.1% of difference is about 3 to 4 million changes in the genome or variants. And what we're doing, we're trying to find the 1 to 2 variants in the genome that are associated with autism and that are causative. And when we did whole genome sequencing in the cohort, we identified about 5 million total variants per genome, and about 200,000 of those were rare variants, and about 130,000 were novel variants, meaning that they have not been reported in other public databases of other populations. And interestingly, about 2 million variants were private in our cohort, meaning that they were not reported anywhere else in all of the other public databases of genomic data. So we did the whole genome sequencing on all individuals in the cohort, including parents, affected children, and any unaffected siblings. And we identified variants in known disease genes as well as new candidate genes. And the type of variants that we identified were both coding as well as non-coding. Coding variants are ones that occur in exons or what we call the exome portion of the genome, and they result in changes in protein sequence. While the non-coding variants are the ones that occur in regulatory regions of the genome, and their functional impact is harder to pinpoint. And so I have some examples listed here of the variants that we identified in known autism genes that are well established. And we also found new genes, but at this point, we need a lot more functional studies and additional human genetic studies to increase our confidence in these variants and new genes. And we then, after we did our standard analysis pipeline, which is this ASD variants that segregate with the phenotype in the families, we used the genomic data that we have to perform something called admixture mapping. What that classical human genetics technique does is it relies on differences in the phenotype based on ancestry. And so the first step that we did for the cohort was to confirm that we have admixture of 2 ancestral populations. In this case, they're African and Eurasian, and this is known about the Ethiopian population. So what you see here is an admixture plot, and we see our East African ASD cohort marked as EA-ASD. And you can see that in that genome, there are contributions from 2 other ancestral genomes, the LWK, which refers to population in Kenya, and the TSI, which refers to the European population, that they are the 2 populations that admixed and gave rise to our cohort. And then we analyzed the ancestry haplotypes under the hypothesis that the higher prevalence in this population may be due to risk alleles that are ancestry specific. So this is admixture mapping. And using this method, which had not been previously applied to ASD genetics, we identified several loci or genomic regions that confer ASD risk. And these loci included known ASD genes as well as potentially new ones. And I've listed here some of the examples of some of the known ASD genes as well. And so, as I mentioned, we still have a lot more to do. We're excited about this study, which was a big first step for us to actually start doing this. And just to summarize our overall work, we established an African ASD cohort for genetics research through this community partnership. And we leverage the ancestral genomic information and genetic diversity in the African cohort that had an unusually increased prevalence of the phenotype to identify ASD variants and risk loci. And when we did this, we discovered about 2 million novel private variants in the cohort. And we identified rare variants in known as well as new candidate ASD genes. And then we did this a classical human genetics technique, but we applied it to next generation sequencing data, admixture mapping. And we identified several risk loci on the African haplotype. We are now actively enrolling to expand the ASD cohort. And then importantly, we're, as I mentioned, really interested in understanding the normal function of these genes in the brain and to pinpoint the specific mechanisms that they regulate. So now we're prioritizing these genes for follow-up functional studies and to investigate the underlying mechanisms of how a specific mutation in that specific gene gives rise to phenotype. In conclusion, we just wanted to share with you some of our, I guess, learnings through this journey is that community partnerships are absolutely essential to drive research in human genetics, especially for underrepresented populations. And that equity in healthcare depends on inclusion of all human populations. Now, as human genetics findings make their way into the clinic, this becomes especially important to ensure that there's no health disparities. And also, our study, like other studies that have been done for other disorders, highlights the power of African genetic diversity to inform us in complex disorders, even when something is as heterogeneous as ASD. We found ultra-rare, what we call private variants, and also the allele frequencies of these variants differ based on the different populations. And then also the importance of using the relevant, knowing the ancestry of your population and using the relevant population allele frequencies to filter against them. So we're excited about the concerted efforts that are ongoing and just want to give a special thanks to the H3 Africa Consortium for providing us with control allele frequency data from Africa so that we can complete our analysis. Our hope is that by identifying the genes that cause ASD and studying the specific mechanisms that are affected in disease, we'll be able to pinpoint targetable pathways that we can harness for the development of personalized therapies in the future. Our ultimate goal is to create this self-propelling flywheel that takes us all the way from gene discovery back to the patient. And it's an exciting time for us to be in ASD research and doing this together. And in closing, I want to thank our audience, all of you, and the individuals with ASD and their family members for participating in our studies and really driving our research. And of course, my partner in this effort, Leah Seyoum-Tesfa, the founder and director of RFASC. And I want to thank the foundations and other family support groups, my team, our network of collaborators, and the funding agencies who support our work. Thank you.
Thank you both so much for a really engaging presentation and really highlighting the true partnership that you all have built together for this work. If you don't mind just stopping to share your screen, we can dive into some questions here. I'll just remind all of our attendees, if you have not submitted a question and you would like to go ahead and find that on the right-hand side of the browser. And we have quite a few coming in, so let's go ahead and jump into them if that's okay, so we can get to as many as possible. So the first question here is, often children with autism are diagnosed in school but never evaluated or tested or referred to medical genetic professionals. Do you have any comments on what is being done to coordinate care between the schools and genetic professionals from your perspective?
I will take a stab at this and then refer to Leah, but I would say that everyone should be referring for genetic testing, because even if the identified variants right now at this moment are variants of unknown or uncertain significance, by doing this, we'll be able to build our full database and full range of variants that are causing autism. So I would say that everyone should be doing this. In terms of how it gets coordinated, I think, unfortunately, there's a lot more to be done there, and there's a big gap. And I think it has to be through the primary care physician or pediatrician to do this, yeah.
I agree that needs to be done. And for the person who asked the question, that's very common that kids are not identified through the medical system. They're only identified in the schools. And I think we've got to do a better job of identifying them by the PCPs and sending them for an outside a medical evaluation for autism. In some cases, they do need to see a neurologist and other autism workup. And I definitely agree PCPs need to do better with that.
And Mountain States has done some work in this area. I told these ladies it was a top topic area for our primary care education initiative, Time for Genetics. So we have a whole module on autism, and we'll be including these ladies' presentation here in that as well. So thank you for commenting on that. We also have a question here. Have you noticed any differences in diagnosis rates between exome and genome? I know you said you were really just focusing on one type, but are you comparing?
So we are doing whole genome sequencing because it's more comprehensive. And even though now it's being treated as an exome, I think, our tools and information is increasing so that we will be able to assign some sort of functional impact to non-coding variants from a whole genome perspective. But at the very least, I think a genome is a more comprehensive exome. So right now, because the genome is still being treated as an exome, and we're only, for clinical diagnostic purposes, we're only zooming in on the exome portion of that. So far, there's no, there's no real difference between the diagnostic rate. We haven't, in other words, we haven't identified a non-coding variant that we would have missed otherwise. Yeah.
Great, thank you for clarifying. And then do you know of any updated studies that discuss the diagnostic yield of Fragile X testing? This particular attendee says, I see a lot of patients with ASD, approximately 10 per week, and have yet to receive a positive Fragile X result. What are your thoughts on offering whole genome sequencing as a first tier testing for this population?
Yeah, I would love to see that. I would love to see whole genome sequencing being offered first, because like I said, it's more comprehensive. I think Fragile X as a single gene, FMR1, has the highest contribution at about 1.94% of cases, but that's, there are other reasons for why that number is high for FMR1. One is because it was being offered as the first tier. So in that sense, the data is kind of biased toward that. But yeah, I think everyone should be getting the whole genome. Yeah.
Great. And then this next question is about any thoughts that you have on carnitine responsive autism. This might be more on the functional side of things, but are you looking at all at things like that?
No, I can't comment on that, but I would say that because we know how heterogeneous autism is, and because the genetics taught us that it's really a collection of individually rare genetic subtypes that may or may not share common mechanisms downstream, I think in general, it's hard to generalize one mechanism for all of them. Yeah, I don't think there's enough data or a strong support for that, for specifically the carnitine.
And this next question asks, do you have any comments on the huge diversity within the African population? Do you foresee similar studies with other African populations?
Yeah, we know that the African genome is the most diverse genome, and that has to do with the history of the human population and population migration and the bottlenecks of that migration through Africa. And I think, we should be doing more studies. We weren't super surprised that we identified all these novel private variants because, again, we are comparing the databases that we're using as our control allele frequencies are largely white European ancestry populations. And so I think we need to do more across all disorders, but also for a control population from the African continent.
Great, thank you for commenting on that. This next question asks, in your experience, have you noticed a difference in frequency of engaging in genetic testing for families who might believe vaccines play a role in their child's autism diagnosis versus families who do not have a concern about vaccines and the role of ASD?
I would refer that to Leah, who has direct firsthand experience with recruiting families to genetic research studies. So for us, yeah, I would say we interact with the families after they've been recruited to enroll them and explain the details of the study. But Leah.
The answer to that question is no. There are families in my group that believe that their children ended up having autism because of immunization, but everybody that was that participated did not really believe that. So it didn't matter whether they believed or not. I think there were some families that were against participating in the study, and some were happily participated, but it didn't matter, because I think a lot of thinking that goes with the vaccination causing autism, it also has some genetic, maybe there's a portion of children that are predisposed to being affected by vaccination. So I think families are trying to find what caused their children's autism, or what is the, is it genetic? Is there part of it? Is there a genetic component to this? So I don't think there was any difference.
Yeah, I think they're interested in finding out the genetics, so regardless. Yeah.
Great, thank you. And maybe just a follow-up question to that. You said there were some people who were very interested and some who just weren't. Was there anything that you could point to that was the differences between families who wanted to engage and maybe families who did not? Any generalizations?
I think there were some people that families that did not want to know more. They were satisfied with what their children's, the autism, whatever caused the children's autism. And there's also some bias as to what are they going to do with the results? What's going to affect? Is this going to negatively affect our community? How were they to find something? There was some concern in that area as well.
Yeah, thank you. Thank you for sharing that. We hear that quite a bit from certain family groups in our work as well. Thank you for sharing. Next, we have a question about, let's see, in any part of the studies, do any numbers point out that the gene predominantly comes from the father or the mother? So specific inheritance patterns that you're picking up?
No. So there's evidence that all types of inheritance modes are implicated in autism. For example, there are a lot of de novo or spontaneous mutations, and these are ones that are not inherited. They arise in the germ cells of either the father or the mother, and then they end up manifesting in the child. So there's a large category like that. There are some that are inherited, but in a recessive fashion, meaning one copy from mom, one copy from dad, and then the affected child has to have both copies to be affected. And so we have evidence for these examples. Yeah. And we did have one rare case of a dominantly inherited situation where there was a variant in an affected father, and he passed it on to his affected children. But those are very rare.
Thanks for sharing that. And I think you may have answered this, but maybe if you could just clarify, this person's asking, I'm curious to know if the study was done solely within the African population, or did it include mixed? For example, African American and Hispanic, African American white, or was it solely with 100% African American?
Yeah, so for this particular study, it was 100% African, more specifically Ethiopian, Eritrean, and one family from Kenya. We do have other studies that are ongoing where we're recruiting all populations, and we have a local cohort here in the Dallas-Fort Worth area where we have individuals who are Hispanic, African American, white, East Asian, and South Asian. But for this particular study, no, everybody was East African.
Thanks for clarifying that. My goodness, there are so many questions we're not going to get a chance to get to. I think this is going to be our last one as we're coming up on time. But this one says, do you think there is any confusion in diagnosis of autism in immigrant children who may have faced trauma in the process of the immigration itself?
So I will again refer to Leah, but I will say that immigration is an environmental factor and stressor for families and pregnant mothers and such. But for our particular study, it was not applicable. And I think, Leah can talk more about this. And also, whether it's a language issue, there are a whole field of study on the effect of immigration as an environmental factor on misdiagnosis. But this wasn't in this particular cohort, but Leah can maybe speak more about specifically immigration and diagnosis.
So there was some of the studies that literature identified mothers having been going through trauma through the immigration process, especially East African from Ethiopia. They go to refugee camps. They do go through a lot of trauma before they arrive to their final destination, let it be United States or Europe. So they did identify that as some of the some possible causes, but the study that we did here, most of the parents were already here. They've already been here, living here, such as mine. When we did, most of them were not newcomers. Does trauma have effect on the children, the ones that have traveled? And I don't think we have any kid that had recently traveled in our study. And a lot of the families that I support are usually the kids were born there in Ethiopia and came here, or they were diagnosed here. They were born and diagnosed here. So our study did not look at that at all.
Thank you for clarifying that. And with that, I will just end by thanking you both again for being here with us and presenting your work and your collaboration.
Thank you.
For more information, visit mountainstatesgenetics.org/contact. This activity was brought to you by Mountain States Regional Genetics Network. Please enter this code in the evaluation survey in order to claim Genetic Counseling CEU, GS23-11.